Now we are going to learn about enzyme catalysis. Already we know catalyst is a substance which increase the rate of the reaction but does not change in its mass and its chemical composition. So here enzymes also act as a catalyst. What is meant by an enzyme? Enzymes are complex nitrogenous organic compound. Nitrogenous organic compound. Organic compound means compound containing carbon and hydrogen. Nitrogenous organic compound means including nitrogen, oxygen, hydrogen, which are produced by living organisms. Living organisms means plants and animals. So what is meant by an enzyme? Enzymes are complex nitrogenous organic compounds produced by living plants and animals. They are actually complex protein molecule of high molecular mass and they are in colloidal state. What is meant by a colloidal? I'll explain in the next video. They are very effective catalyst. That is, catalysts numerous reactions, especially connected with the natural processes. Numerous reactions occur in the bodies of animals and plants to maintain the life process. These are all catalyzed by enzymes. So the enzymes are termed as biochemical catalyst. The phenomenon is known as biochemical catalysis. Enzyme is a biochemical catalyst. The phenomenon is known as biochemical catalysis. So many enzymes are obtained in pure crystalline form from living cells, the living plants and animals. However, the first enzyme was synthesized in the laboratory in 1969. Now we can see many enzyme catalyst reactions. What are the enzyme catalyst reactions? Number 1, cane sugar C12H22O11 on hydrolysis in the presence of an enzyme invertase. Enzyme invertase. This cane sugar breaks into glucose C6H12O6 plus fructose C6H12O6, glucose plus fructose. This is also called as inversion of cane sugar because in the presence of an enzyme invertase. Number 2, glucose C6H12O6 in the presence of an enzyme zymase, it is converted to ethyl alcohol C2H5OH plus carbon dioxide gas is evolved. So conversion of glucose into ethyl alcohol in the presence of an enzyme zymase. Third one, starch. Example C6H10O5 N times. Starch, this on hydrolysis in the presence of an enzyme diastase. The starch converted into glucose C12H22O11 maltose. The starch converted to maltose. Conversion of starch into maltose in the presence of an enzyme diastase N times. Maltose C12H22O11 on hydrolysis in the presence of an enzyme maltase. It is converted to glucose C6H12O6, 2 molecules of glucose. Next, decomposition of urea formula NH2, CO, NH2, 2 amine group. On hydrolysis, in the presence of an enzyme, urease forming ammonia and carbon dioxide. Ammonia and carbon dioxide. Decomposition of urea into ammonia and carbon dioxide in the presence of an enzyme urease. Then sixth one, in stomach, the enzyme pepsin. The pepsin enzyme converts the proteins into peptides. Proteins into peptides in stomach. While in intestine, this is in stomach, but in intestine, the pancreatic trypsin converts the proteins into amino acids. Proteins into amino acids by hydrolysis in the presence of pancreatic trypsin. Now, next, the conversion of milk into curd. Milk to curd. This reaction is brought about by lactobacilli enzyme. Lactobacilli enzyme present in curd. So, these reactions takes place in the presence of an enzyme. So, this is called as enzyme catalysis. This is also called as biomedical catalysis. So, these enzymes are called as biomedical catalyst. Many reaction in our body, it is stomach, intestine, many enzymes are responsible for functioning, digesting, etc. So, the enzymes are very useful for our life because it is produced by living plants and animals. They are more useful in only the natural process. This is given clearly in book, page 133.